All right, so basically today I was hoping to do a little bit of review if we need to for the test for Friday, right? So the short, we just have two chapters, chapter 6 and chapter 7. Right. I would expect it to be fewer multiple choice questions and then some writing. Right. So I would say chapter 6 kind of is broken down into, we've got tools. So how to study cells with the homogenization and microscopes, and then just marching through all the organelles, structure and function, each of the organelles inside the eukaryotic cell. It is, but it isn't, right?
Yeah.
So
it's a lot of information, but not a lot to.
right, it's not a lot of process things. It's a lot of memorizing things, right? And keeping things together.
Yeah.
So like, if you can remember 5 things about the mitochondria, you're fine. And the like if you can remember all the things that we had on the outlines, you'll be fine. Chapter 7, same, the beginning, structure of the membrane, and then the process part of that transport. Basically, that was the rest of the chapter: passive transport, active transport, and the different types of each. That's pretty much all we did, right? So definitely multiple choice, maybe some matching. I doubt it. Usually no.
They don't know homework.
Oh, that's because I just
Yeah.
I wanted to make you do that. So that's left over from COVID times when we weren't in person. And so I didn't have that scientific that scientific vocabulary lab for people to do. And that was on the mastering. And I was like, ooh, then I won't have to upload that. Click, they can do that homework. So
I want the homework and learn to get quiz chapter 9 or something on Saturday.
probably. So that's to try to force you to start to get through that material. So let me put that on my to-do list. That should probably be a little later. It shouldn't be for this Saturday. I haven't had a chance. There's still a couple people that are having trouble with their access to connect. Are you in now? Fine, and it doesn't bother, but James seems to still be having trouble, and I don't know what he's doing that, because he's the only one out of this class and my other in-person class and my online class that is still
Well, we were having problems. We sat in the cafeteria, and then eventually, when it came back up, mine worked, and then I guess he tried.
Yeah, if he's trying to do it here, that's probably the problem, because the internet here is not. It's not like if your internet at home when it's just 3 or 4 people on the internet. You know, it's all of us are in the portal.
If I want to work it out, because it's just me on it.
Do you know the date on it?
9.
9th? All right, I'll move that. That's a mastering move. That's fine. I'll just move that. Literally, I had planned to go in there today and futz with where we are for your online assignments. For the next chapters will be 8, 9, 10.
Yeah.
That's the next set. And so then I'll probably move all those deadlines, all those due dates to right before the in-person test.
So you don't this for 9's on going to be Friday then?
No, I'm probably not. No. So that would have been if we were still like, when I made out the schedule, I made it out for the Monday, Wednesday, Friday for you for Monday, Wednesday, Friday, and I just can cover so much more ground when I see you 3 days a week for shorter periods of time than when I see people 2 days a week for 3 hours each. It's just a matter of attention, you know, like literally, even on a Friday, if we have a lab that's going to take an hour and I talk to you for 40 minutes of the last bit of some lecture, we're still out here for 2 hours, you know, and we get all that done in a day, but it's really hard to get all that done with them in a 3-hour block. So yeah, they're brutal. Yeah, even if we do lab, and it's later in the day too, so everyone's a little more restless. You guys are a little more docile. 8 o'clock in the morning, I can usually, it's easier to, I don't know, it sounds bad, but it's easier to control the room and like force the material than at 2 in the afternoon. I'm tired then too. Right? The other thing that I wanted to talk about for 6 and 7, and this is important for the people at home as well, is for the short answers, because I would like to practice a little bit with you guys. I would like us to start to practice writing a little bit more on our tests. It is an important skill, especially for any of you who are going to continue in science or even in allied health. It's a way to practice better communication, right? And so, like I'm always telling you, in Bio 1, we're trying to teach you how to do all these things, like physical skills of learning to use the microscope, but all these soft skills of being part of a team, sharing information, learning how to write a little bit better, more concisely, more scientific. Even if you're a nurse and you're going to be typing notes, the clearer and the less language you can use to communicate, the clearest thoughts are going to get your patients the best care, right? Because no doctor wants to read a long narrative of what happened to that patient that day. They want to see the meat, but what is the important parts? And so this is a training to try to help you get better at that. So always for the short answers, those are usually what we consider higher learning levels of Bloom's taxonomy. So it's usually knowledge plus, right? So it's either plus description, explanation, or a critical thinking exercise. Or sometimes you'll see it called analysis. I don't usually do the analysis ones too often in Bio 1. Occasionally I will, like, so for example, we did the lab about the potatoes and osmosis, right? So sometimes I will include on this test, one of the short answers will be a small set of potato data or a graph of potato data or cucumber data. Maybe I'll use a different vegetable. And I will ask you to interpret that data and tell me what is isotonic for this piece of fruit or vegetable, and how do you know, right? And that would be more like this, right? It would be taking the skills that you learned in the lab and then applying them to sort of a new situation, right? Probably that would be an extra credit on this one, okay? Most of the type of short answers that I would ask you in this particular test would be things more like the following, right? Describe 2 different types of microscopes. And be sure to include their application or what they're used for. Now, what I would be looking for, obviously, right, and what they're used for. So what I'd obviously be looking for, that's very open-ended. We talked about compound bright field microscope, compound phase contrast microscope, fluorescent microscope, transmission electron microscope, and scanning electron microscope, right? So we talked about 5 different types of microscopes, really. Pick any of those, pick any 2, and why don't you try it? Do it. Ready to give me an answer? At least like an out loud answer, right?
So do you want me?
So start with this one. So give me 2 different types of microscopes. Tell me which 2 you picked.
Okay, so I picked the transmission electron microscope
Okay.
and I picked the compound bright field microscope.
Okay. So TEM and compound bright field.
TEM uses a slow specimen or concept in it.
Right.
So you see inside cells or the non-bacterial virus. Is that good?
That's good. It would be a key to tell me the magnification of that,
Okay.
right? So you've given me the application, right? So the application is to see inside cells, to see viruses. How much does it magnify? It's easy to remember. It's easy, 100,000 to 1,000,000 times the size of light. You just remember electron microscopes as big as 1,000,000 times the size of light.
When the compound bright field, it has the 4 times 40. And if you do the objective lenses.
So what might be, so like to condense that a little more, might be compare them directly with their magnification. So if you're using that 100x objective or even the 40x objective, what's the magnification on my compound bright field microscope then? What's my max? Yeah, 400x. So if you put that, see how that's like direct
Yeah.
focus.
And shows you how the bright field's a lot, it's not as strong. as
Yes.
the
Yes.
TEM.
And you're giving me the detail to show me that you know that they're the reason that they're not as strong, that you can't magnify as much. So, and then what kind of cell, what kind of things can you see with that? Or
I guess I have to take any cells or skin or blood.
so you can see cells, and what structures of the cells could you see with that? Cell walls and what else?
Cell membranes.
The nucleus, right? There you go. So you can see the edge,
And the-
right? The boundary, the membrane or the cell wall, and you can see the nucleus, which is the largest organelle. And that clearly illustrates that you know that with the electron microscope, you can see a lot more detail and things that are a lot smaller, like an organelle, right? Or a virus. And that way you've given me exact comparisons. On the one thing I would also include, so this one, you told me you slice it and you see things in cross section. I would say here that we stain specimens.
They don't stand for the other one.
Right, otherwise you can't see them.
You can't really see it.
Right, so now you've given me things that are directly different or similar between the two microscopes. And reasons.
Well, description.
And
not given too much.
Right, and very specific, right? I usually tell students, try not to put it just as a list, try to do this and then write it into sentences. Because again, we're working on organizing what all the information that you have in your heads for this chapter is in there, just swirling around like crazy. And part of what taking a test is about is figuring out which pieces to pull out. And then when I have those pieces, how can I organize them best? Right?
For the scanning electron one, is it the same magnification?
It can be, and I wouldn't like, since we're in Bio 1, if you wrote 1,000,000 times, I wouldn't mark that wrong at all. The key difference between the scanning and the transmission is the seeing into the cell and seeing the surface.
Sorry, I have to go. I got my brother off.
That's fine. That's fine.
My parents just texted me the way.
No, that's fine. I'm recording, so after I'm finished, I'm gonna sit in my office and try very hard to upload it to YouTube. It's a technical thing. Sitting at my desk, my phone, like my verification into YouTube is type this number on my phone, and my phone doesn't pick up the Wi-Fi signal in my office unless I'm up against the window. And then then it's like, you need to put your password in. I'm like, I don't know my password. And then I just get mad and stop. So, but I will try to do it today. Yeah, let's keep going because I'm glad you came, Sophia, but it's not just for you. This is supposed to be for everyone who's not here as well, to help them do better on this portion of the test, right? So that was excellent. This is a question that I often ask, literally both of these. I usually don't ask the microscope one and Bio 1, but I probably will put it on there. Always bear in mind, if I'm going to ask you guys to answer 2 of these. There'll probably be 5 sitting there for you to choose from. Okay. So if one of them, if you're just like, I don't know the answers to this, don't do that one. Leave it blank. Yeah. Right. Just pick out a different one. Okay. So you like this one? Yeah. Okay. So compare and contrast. That's usually a little bit of what I just erased with the Bloom's taxonomy. That's a little more of knowledge plus tiny bit of, can you do a little analytical thought, right? So for compare and contrast, what I always tell students is it's worth 5. And the rubric for all my compare and contrast, no matter what class I'm teaching, it's worth 5 or 10 or 6 or whatever. The rubric is always, tell me one thing that you know about them that they share in common. Tell me one thing that why these 2 words are put together. Why would I even ask you about these 2 words? And in this example, it's kind of easy because obviously they're both about transporting molecules across the membrane, and it's basically in the question, right? If I would purposefully choose something very disparate, like compare and contrast, not the chloroplast and the mitochondria, because those are very similar to each other, but compare and contrast the mitochondria and the nucleus, right? Then I'm asking you for a little more analysis. Like you might have to think for a minute, well, what can I say about those that's the same? They both have DNA, they're both organelles, right? So give me one thing that they share in common, and then basically give me definitions, right? And each definition is worth 2 points. So you're getting a point for telling me when you're writing your answer, you're getting a point for telling me a truthful thing that they share. And then you're getting 2 points for the definitions. And in this case, since I asked you to include an example of each, I'm probably going to put a half point on each of those examples. So the base definition will be 1.5. And if you can say diffusion or facilitated diffusion for passive transport, that's going to be that extra half a point. Does that make sense?
Oh, you want me to say?
Well, that would be
Okay.
nice. That's good
Okay.
practice. So what are the two big things? Go ahead, say.
Well, the same, like the compare is, they both move things over the plasma membrane.
Good.
And then passive transport, there's no energy, and it moves from a higher concentration to a lower concentration.
Very good.
And then for active transport, we need energy,
Mhm.
and they move against the concentration gradient. And then an example for passive would be diffusion, and then for active, could I put like the type of pump, or
Sure.
so like an ion pump?
You can put an ion pump, or you could put sodium potassium pump. Yeah. Yeah. The more things that you can tell me that are true, the more points you get. Right. And I've also been certainly known, like if you really know all those things and you had all those things in your answer, and even if you said specifically, I don't know why you said specifically for passive transport, if you said diffusion, like gases, oxygen / carbon dioxide, I probably would, and you could do that for the other one, I probably would actually give you 6 points. I mean, if you know it and you can clearly explain it, you get all the points or more, depending on how much you gave me. Okay. This one is one of my, I always ask this one. What organelles will be used by a cell to synthesize a lipoprotein? Think about that for a minute. No one ever answers that. Don't ever fix that one. Well, maybe you would now if you think about it ahead of time.
Yeah.
Right? And then we'll talk a little bit about what to do to figure out, like, what am I going to ask on the test? Because it's not rocket science. It's really very calculated. Oh yeah, use your notes. That's the whole point. That is a hard question, but I'm almost positive that they asked it of you in the homeworks, and I know that there's a picture of it in your book. Specifically after the Golgi section, I know there's a picture of it in your textbook. Golgi will be a part of it. So I'm going to put that on my, like, over here on my scratch list of what I'm going to include in my answer, right? The Golgi is definitely a part of it. And what before the Golgi? What organelles actually synthesize the protein? What's made in the smoothie are? Yeah, and what else?
It synthesizes phosphorus.
And other lipids. Okay, so we're going to use that smoothie R is going to be involved because that's where the lipid part is going to come from. Where are proteins made in cells? Ribosomes, which are on the Rough endoplasmic reticulum, good. ER. Rough because there's ribosomes, good. That's where the proteins are being made, and they're being shoved into the rough endoplasmic reticulum because that's what happens to them in the rough ER. I'll help you. Primary and their secondary structure, right? So when the protein is made, that chain, that list of amino acids is what is being, as soon as those start to be synthesized on the ribosome, they get pushed into the endoplasmic reticulum. And that chain just grows and grows and grows and grows until it's that 200 amino acids long, like thinking about spider web protein, right? That would have like 125 amino acids. Then the secondary structure of where any part of that polypeptide chain that's going to form alpha helix or beta pleated sheet, that happens in the ER. And then tertiary and quaternary in the Golgi. So then it's going to go to the Golgi. We're going to get any tertiary quaternary structure, plus that's where the lipids are going to meet up. Right? That's where the lipids are going to get put on their head of the protein. And then we need one more thing. We need vesicles to transport this around from the ER to the Golgi, from the Golgi to the Golgi, and then wherever it's going to go. The smooth ER to make the lipid and transport it to the Golgi. And we actually also need the nucleus because that's where the code is to make the protein, right? That's where the DNA is to make the RNA to make the protein, the sequence. That's a really hard one. I know I never answered it. But like I said, there's a whole section in your book that's called like the endomembrane system or something. And I think it comes right after the Golgi because they're trying to get you to see how things are connected. Like I said, that's a really hard one. And it isn't something that we focus on in class, but if it's something you know from before, or if you took the time to read that section and you're like, oh, this is how this all goes together, if you bothered to figure it out, then you're prepared to answer it. That's like a serious synthesis question. You have to take things from all different, not just memorizing everything in its little cubicle, but then seeing how those are all connected. Like I said, since we completed the osmosis lab, which sometimes we aren't completed, we haven't talked about what to do with the data yet when I give this test. But since we've completed it and we talked about how to make the graph and what it means, hypertonic, hypotonic, isotonic, I'm inclined to include that question on in the short answer. So a piece of data, maybe a picture of a graph or a similar vegetable, not a potato, but a cucumber or a radish or something. And I would say this is data collected from an experiment similar to the one we did in class. Here's the line. What's isotonic for this vegetable? How do you know? Or might ask some questions about it. I had already mentioned that. I just erased it. I had already mentioned that before you walked in, so it's on the recording. But okay, since you guys are here, you probably won't watch the recording. Which I
still watch it.
Which I get.
I like watching while I'm driving. I listen to the records while I'm driving.
It's frightening, isn't it? That like my voice is just like being projected over. I made a podcast of a microbiology lecture in like 2004. And at that time, the equipment that we had, it was no visual, it was just an audio. And I had to go and sit in a little like soundproof room that the college had to do it. And I made this podcast, and at the time, my students were just like, it's so cool. You're like coming out of the radio of my car. I'm just like, yeah, that's kind of frightening, but whatever. Now it's commonplace, right? But at the time it was kind of a weird new thing. Good morning. We're done. No, I'm just kidding. We are, but. So the last thing that I would say to you guys that I'm going to put basically on the recording is that I would remind that all of the exams that you're going to take from me are always about 65 to 70% knowledge-based questions. They are basically, do you know the stuff? Do you know these definitions? Do you know what these terms mean? And don't forget that I can ask you 5 things at a time, right? So amongst this audience, so I'm recording it so you can see this later and we can go over it again after I stop recording, but I need space to write. So about between 65 and 70% of all of the exams or lecture quizzes that I give you are knowledge based. And so that's like the basic level of Bloom's taxonomy. Do you know the material? And this, I understand, is a bit challenging for us now because we've all lived through a virtual world where you may or may not have had to know anything because they gave you too long to answer the questions and you just looked them up. And no judgment, right? No judgment, but that's not an option for science. You are going to sit for a nursing exam, or you are going to sit for the general record exam, for the GRE exam to go to graduate school, or you're going to sit for the MCAT exam, and they are timed knowledge questions. Do you know this? Can you analyze this? Can you answer this question? So it's not helpful for us to continue that, right? And I understand there's going to be some growing pains with it, but I can't just say, oh, use your notes. That's not a test. That's a homework assignment in class, right? That's who has the best notes. Right? That's not who knows the material. That's who knows how to organize the material, which is important, but isn't the whole thing, right? So, like I said, the majority of the exam is knowledge-based. Do you know the material? Because the object is, you know, a C in a course is average. Do you know the material? Do you know most of the material? More than half the material, right? Do you just know it? So the difficulty is for beginning students, though, is like in chapter 6, I can ask you, like I said, I can ask you 4 or 5 knowledge-based questions in one question, right? So if I use a multiple choice format, and people will always, you know, they're always like, you didn't ask anything that I studied. And I'm like, okay, how is that possible? This is a test about chapters 6 and 7. In a multiple choice question, for example, I could ask you about the structure of 5 organelles in one question. Which of the following organelles is incorrectly matched to its structure? Mitochondria. Double membrane with inner membrane folded into cristae. Chloroplast, triple membrane, right? I could ask you vesicle. I could, any one I want to pick of the 8 or 9 organelles that we discussed. Or which of the following cytoskeletal components is incorrectly matched with its function, right? And I'm already asking you 3 things, 6 things you have to sort in your brain in one question. So I don't need 150 questions to ask you all this material, even though it seems like there's thousands of things for you to know. I can compress it very quickly. So it's important that you spend the time to read the questions carefully, which when you're anxious, you don't do, because you just want it to be over, right? So part of the managing the anxiety of taking a test is at being prepared for the test. Everyone's nervous when they take a test. It doesn't matter who they are. Everyone's nervous that when they take a test. Only way to mitigate that nervousness and anxiety is to really know the material. Because if you really know it, then you're going to be confident when you make the choices of the answers of the questions. It's not any harder than that. Unfortunately, because it's biology and there's a lot to memorize, that takes hours and hours and hours of preparation. It's not psych. You can't just go like, oh yeah, that example she talked about, that's what's gonna be on the test. I just have to write like 3 sentences about it. No, it's not like that, unfortunately. So there's a lot of stuff, there's a lot of work that goes into, do you know the knowledge? And then the rest of the test. So these are all this, what I call the C questions, right? If you get all of those right, or most of those right, you'll get about a C on the test. Magic. Then there are the B questions, right? If you get most of the B questions right, not choosing the letter B, which students often misinterpret when I say that, but choosing the questions that are more about analysis, right? Or more about explain, or more about, actually more about process, usually about list the steps. Like that's what I think of when I think of a process question, right? Which of the following is the correct order of events if if a scientist is trying to purify ribosomes for study under a microscope? And then the choices would be about homogenization and differential centrifugation to fraction out those parts, right? And you have to pick the right one. So the knowledge is you have to know what that process is, and then you have to pick through which one is the best choice. Then there are the A questions, right? And the A questions are the harder ones. Those are the critical thinking questions. Really, those are more of the analysis questions. These are more explain, list the steps. So Sophia, like this question, right? What organelles would be used by a cell to synthesize a lipoprotein? This would be a question that I would use either as a short answer question, see what you know, or I can turn that into a multiple choice question. And that's an A question, definitely, right? Which of the following list of organelles is the correct order for a cell to synthesize a lipoprotein? And then, you know, there would be A, nucleus, rough ER, smooth ER, Golgi. B, you know, nucleus, mitochondria, you know, whatever. I could make up any combination, or I could mix up the order of them. So I think one of the, when you're studying, so the beginning part of when you're studying is figuring out what it is that I have to know from all this material. There's always going to be more in the book than we're going to go over. That's the whole point of me, right? If it was just about, can you memorize the book, you wouldn't need me. My job is to tell you, these are the things that are really important, or these are the things I think are really hard for people to understand. So I'm going to try to explain it to you, right? That's why you take notes. The notes are your review sheet. That's what that is, right? You don't need a study guide, you have one. We've spent the last 2 weeks making it, right? Then you need to spend all the time, which it's best, of course, if you spend the time in small increments every day, studying just a little bit of it, because an hour of this at a time is about all most people can take. Sitting down, I know it's already Wednesday, the test is Friday, and I know there's only 2 chapters, so it's easy to think, oh, I can just do this 1 chapter tonight and 1 chapter tomorrow night, which is true. Then I see you in this area here, in this C, maybe B area, right? If you're really good at memorizing stuff, yeah, you can probably do 2 chapters of this. If you've had it before, you can probably do that. For most people, this is a, you know, siege or a slog of daily little bites of things that you don't know about or aren't interested in or don't want to do, but force yourself to do in small increments. And then at the end, you can pull it all together. The best is if you have an hour at the end to think about, okay, I'm looking at the questions at the end of the chapter. I'm looking at those some of those dynamic study modules I've attached to the chapters in mastering, or I'm going over the homework. And now I'm like, oh, that's why the answer was B, right? That's hard to do. I mean, I'm not going to lie to you. That's really hard to do, to have enough time that you feel like you've studied all the material, and then you have a little time left over to look for the connections between the material. Doesn't happen super often in Bio 1, and I don't expect it to. I expect it more frequently in the 200 level courses, and so will your instructors. Okay? I'm not going to ask you questions with multiple answers. There are never purposefully multiple answers on a test. That again, that's something more advanced that you'll need to practice, because on the nursing board exams, that's how they are written. They're like, choose all the answers that are possible, right? And if you miss one of them, you get the question wrong. So it just gets more difficult as we go along. I'm trying to, again, in Bio 1, teach you some skills that will help you take a lot of tests, not just these tests, right? The writing portion we already discussed before you guys came. The writing portion, you're gonna write 2 short answers. I'm gonna put 5, probably 4 or 5 on the test. And the purpose of that is to help you start to communicate complicated information clearly and as concisely as possible, right? Because as we were talking about before, if you're a nurse and you need to write your notes, no doctor wants to read a long paragraph narrative about what happened to the patient today. They want to know what are the 2 or 3 most important things that you saw today, right? And that you addressed today. Not, I came in and I washed my hands and then I looked at the patient and they looked a little gray and then I did that. No, right? So when you're writing the short answers, again, this is all about practice and training you to think about things. You have all this information, what are the most important parts and how can I put it together in the most logical way? Okay. So that's my advice for the test. I'm going to stop recording.